Hello. This video is meant to serve as a kind of a statement of purpose and hopefully a demonstration of what I think technology like mixed reality can bring as a tool to augment or supplement our methods for communicating and demonstrating and visualizing concepts in developmental biology. So first, let me start by defining what mixed reality is. You may notice there's something on my face and some things in my hands, and that I'm not actually coming to you in a real space. And that's because I'm currently in a virtual reality environment. And the reason you can see this is that you're looking through a composite of two different views. You are looking through the view of a webcam, which is pointing at me, and that webcam is showing you myself. And the picture of that webcam is being composited with another view, which is from the virtual world, from a camera that's physically calibrated to the position of that webcam and is giving you the same perspective in the virtual world and compositing the background and the foreground of this program called Tilt Brush. And so mixed reality essentially mixes the real world with the virtual world and allows you to see basically everything that I'm doing in this virtual space and with all of these two virtual tools that I have in my hands. The reason I think a technology like mixed reality can be used either to uh, record demonstrations or lectures, or even to give live or demonstrations and lectures that are augmented with digital media, is that I think we are sort of limited in the way we can communicate biological concepts and developmental biological concepts in specifically. And this comes from a kind of a dream that I had as I was coming up as a college student. I loved lectures. I loved learning from my lecture components, and I was a big fan of Carl Sagan and his Cosmos series. And I always wished that one could give a lecture with the same kind of media that you had in a series like Cosmos, the special kind of 3D media that he used, but on the fly as one would use a chalkboard. So to be able to come up with a concept and draw it on the chalkboard is really great, but to be able to make a 3D digital image of, say, an embryo would be even better. And I think a technology like mixed reality can allow us to do that. Um, and specifically, this is important because we have a problem when we're discussing biological concepts, especially developmental biology. The problem is when I'm speaking to someone, I am talking about molecular events that are happening in a certain place of an embryo or a certain place of a cell, even within a tissue, which is within an embryo, and happening at different times and all over the and is involving movements and shape changes. And my ability to communicate that to someone relies on their ability to visualize what I'm telling them. And right now, the best technology we have is PowerPoint, which is great, but I think it's limited in a few ways. Firstly, the major limitation is that when you give a PowerPoint presentation, you have digital media, but it's usually projected somewhere away from you. It's on a screen. People are constantly, the problem with PowerPoint is it's inherent to it that you have to ask your audience to split their attention. They're looking at images, they're usually reading text while you're speaking to them. And it's in two different places. You're usually off in a corner, hidden in darkness behind a podium, and your data is over there and your figures are over there. People are looking back and forth. Should they listen to you? Should they read what you have on your slide? Should they spend time interpreting your images and graphs? This is a bit of a taxing thing to put on to your audience, especially if, say, they're undergraduates and are unfamiliar with the material itself. As a biologist, I've had years of experience looking at biological images, understanding schematics, and reading data slides. An undergraduate, for instance, is not going to be so intuitive. It's not going to be able to intuitively read those images as well. And also, if I'm going to collaborate with someone outside of my field, if I'm going to go to an engineering department or if I'm going to give a seminar to mathematicians or physicists, I can't rely on their ability to look at a picture of an embryo or histological slide or a schematic of molecules, things that they're not used to seeing, without me being able to simplify it and manipulate it and show it to them in a different context. If I take it out of the context, slap it onto a slide, it's kind of difficult sometimes to interpret. And this can lead to some miscommunications. It can lead to perhaps collaborative efforts not leading to the fruit that we would like to see. And so I would like to first give a little demonstration of what I'm talking about. And so what do I mean by being able to have a chalkboard in 3D? Well, this program, this is called Tilt Brush. It's by Google. And it allows me to basically paint in 3D. And it also allows me to control the objects that I bring into it. So let's demonstrate for just an abstract example. Sometimes I might be talking about a pair of molecules. Usually we represent proteins with kind of these squiggly diagrams. And let's say we talk about two proteins and they interact with one another. Okay? And this interaction is usually happening in the context of, let's say, the nucleus of a cell. And that's kind of like a ball like this. Okay? And then that nucleus is in itself is packed inside of a cell. All right? So that cell is kind of, maybe it's a square shape. Maybe it's an oval shape. Cells can take a lot of different shapes. And that cell itself is going to be part of a tissue. And that tissue is going to fold and it's going to undulate. It's going to go in many different directions. And then, of course, that tissue itself is going to exist in the context of an embryo. And so if I was using something like PowerPoint, right, I wouldn't be able to show you this embryo. I wouldn't be able to zoom into that embryo or start peeling away the layers of that embryo to show you the tissue and to peel away the tissue and to show you the cell within that tissue and then to go even farther into that cell and now come over to the nucleus where our two molecules are interacting. And I can even, get rid of that, I can even grab one of these molecules, just make it as big as I want, turn it around, and we can see it in all different directions. Okay? Now this is just a very abstract demonstration. What I'm going to do now is load up a couple of scenes that I've prepared. One with a model of a fly embryo or a fly larva, which is actual data. And I'm going to talk about that a little bit, and I'm going to move it around and show, demonstrate it. And then I'm going to have another scene of an embryo that I've drawn with this system, and I'm going to show a little bit of a certain event, a very specific event in embryogenesis that's important, and how we can talk about how different parts of that event take place in different parts of the embryo. So that was the beginning part. Now I will load into the other scenes. Hello again. So what I have done now is I've loaded a scene in Tilt Brush into which I've brought this model. And what is this? This is a model of a larva of the fruit fly, Drosophila melanogaster, that I've created with data that I got from a micro CT microscope, which is X-ray tomography, and allows you to scan a specimen through and through with X-rays and reconstruct the 3D structures of inside and out. And what's really cool is now that I have this model, which is now a true to life representation of what's inside this animal, I can do a lot of cool things. I can first show you the outside of it, I can move it around here. I can show you this beautiful face. We can look down into the mouth. There's sensory structures all through here. The mouth is down here. I can rotate it around and talk about the external anatomy of the larva, the different segments. But more importantly, I can cut this model in half, and then I can open it up to show you what's inside. There we go. Yeah, and we can see a lot of cool stuff happening inside. Now, this entire region is the head of the larva. Okay, we've got a big structure that's specific to feeding, because the larva, it needs to get a lot of food in, it needs to grow and accumulate enough calories and mass to make an adult fly. And then back here you have the brain and the rest of the body. Now, why am I talking about the fly? Why am I talking about Drosophila melanogaster and showing you a model? Well, that's because the fly itself is a model organism. And what does that mean? A model organism is an organism that sits in a kind of a sweet spot of complexity where it's complex enough to exhibit the kind of phenomena that we biologists are interested in because we're interested in understanding our own biology, our own development. We're interested in understanding the genetics and the molecular mechanisms that make that development happen correctly because we're also interested in understanding what happens and how when those mechanisms go wrong, we get diseases such as cancer and developmental disorders. And so the fly is complex enough to exhibit those phenomena. And thanks to our evolutionary relationship, the same genes that are responsible for those phenomena are the genes that make those phenomena happen in us as we develop. And so we can study the fly because it's complex enough to exhibit those phenomena, but it's simple enough to be accessible to our experimental observations. It's simple enough that it's amenable to genetic manipulation and to molecular manipulation. So it's in that sweet spot of complex enough, but not too complex where experiments become untenable. Okay? So what's one thing I can focus on this fly? Well, I have this whole structure here, and you can see that there's a part sticking out, and I've made it stick out on purpose because I've isolated it from the rest of the animal, and I've made a specific model just for it. And so I'm going to clear some of these away. See ya. There we go. And I just want to talk about this structure right here. And this is called the cephalopharyngeal skeleton. Now, it's a skeleton because it gives structure to the inside of the animal. It's a place where muscles attach and different organs attach and get structure. It's cephalopharyngeal because it's part of the head, which is the cephalon, and it's also the feeding structure. So the food enters through the mouth hooks, comes through this tube here, and enters the digestive tract. Now, what's very interesting about this structure is that the very same signaling mechanisms, the very same cell movement and cell shape changing mechanisms, and the same genes underlying those mechanisms that made that structure also made this one. That's right, my face. A lot of the signaling mechanisms that are involved in making this shape correctly are also involved in forming the facial structures. And when you have problems with the development of these structures, in humans, we can have problems like prosencephaly, holoencephaly, cyclopy, which are very nasty developmental diseases. And we can have very bad developmental problems in the fly as well, but it's the fly, right? So we can try to mimic those kinds of developmental defects with genetic perturbations. And let me demonstrate first what I mean by how amenable or how observable this structure is. And so what I'm going to show you, let me just turn this way first. There we go. Up. There we go. So here is an image of an actual fly larva, right? And what I've done with this larva is I have rendered all of its soft tissues optically clear. And so all you can see are the hard cuticular skeletal structures, which is basically this. And so you can see a lot of the different structures are mirrored here. And this is a very simple procedure and very easy to observe on a microscope. And it's great because of that. So what happens when we cause molecular perturbations, when we mutate genes responsible for making this structure? Well, we can actually make a lot of different changes to how it looks. And so here's one example. In this example, these structures here, all of these structures in the back have either collapsed or they haven't developed correctly. And so now you have this big bunch here, right? This whole bunched up here. The front is all right, but just the back of it's messed up. And let me give you another example. Another example, which is more extreme. Now we've taken away a lot of those structures. And so you can see they're even missing. It's not that they're malformed, but they're actually the cells that were supposed to make those structures just weren't there. And in an even more extreme case, we can have it completely gone. All right? So this is an example of a structure that's accessible to experimental observation and also accessible to manipulation. Now, what is the specific process I'm manipulating? What are the problems that I'm introducing? And so I want to demonstrate what that process is. I want to demonstrate how we can mess with it with a different scenario where I've drawn an embryo at a special stage of embryogenesis where the cells that give rise to these structures are specified. Hello again. So what I am showing you now is a different kind of representation. And so this is the same kind of drawing that I made in the previous introduction part of this recording. And so what I've done is I've used the paintbrush tools and I've gone in and I've painted this whole scene, let's say, and I'm going to take us through this scene and see what it can tell us about this embryo. What I want to show you at the outset is something that we can observe just from the surface, okay? We can see that this embryo has some shape to it. Interestingly, at the top, it's kind of flat. At the bottom, it's more curved. And on one side, we have a kind of appendage coming out. And that appendage has a specific name. It's called the dorsal appendage. And that's because it occupies a specific space in the embryo. And so there are two different axes along which this embryo is already patterned. And we have the dorsal to ventral axis, which is why the dorsal appendage is up at the dorsal side. And we have the anterior to posterior axis. And we can see that the dorsal appendage is at the anterior side of the embryo on the dorsal half. And that's very interesting because that tells us that when this egg was formed, it was already formed with a little bit of information stuck onto it. And that information is read out on the outside of the embryo itself. It's read in the differences between the dorsal and ventral side, and it's read out in the differences between the anterior and posterior ends. Now that's very important because we have to ask the question regarding that cephalopharyngeal skeleton. How did the cells that make that structure know what they needed to be at the beginning of embryogenesis? And so I'm going to take us onto the other side of this embryo and show you inside the event that took place that made that happen. So I'm just going to rotate this around. There we go. And let me just find the right position for this. Here we go. All right, so here we have the embryo. All right, and what I want you to notice is that there's some things happening on either end. Now, this is a very interesting stage of a development that's specific to the fly embryo which is called the syncytial blastoderm. Okay, so syncytial meaning same cell, because all of these nuclei, these blue nuclei, they're all occupying the same cytoplasmic space. It's a blastoderm because all of these cells now have arranged themselves onto a cell layer. So they're all in a layer on the periphery of the embryo, but they're all sharing the same cytoplasm. Okay? Something is going on here at the ends. We can see there's some material here on the anterior end, which actually corresponds to the anterior dorsal appendage. Let's look a little bit closer and see what's going on in that region. So just gonna bring this over here, and I'm gonna zoom us in. Okay, there we go. Now, I want to point out something very interesting here. There's 3 colors, and these are 3 different layers that are part of this egg. The outermost layer is the chorion, which is the eggshell. Okay, the inner layer is called the vitellin membrane, and this is a membrane made up of a protein called vitellin. It's hydrophobic, and it serves as a kind of liquid barrier to the outside world. So the embryo itself does not dry out. It allows oxygen through the dorsal appendage, and it keeps in liquids. What it also has is something that was deposited by the mother, and that something is actually information. It's information that tells the cells at this periphery what they ought to be when they grow up. And what's going on here? How does that information that was deposited by the mother on the outside of the embryo, how does that information get into this embryo? How is that information interpreted by the cells? And let's have a look a little bit closer and see what it takes for that to happen. So I'm going to zoom in a little bit farther. Okay. And I just want to focus on the outside of this embryo. Okay. So now I'm taking us into that paravitellin space. And we can see we're surrounded by kind of these blue particles. Okay. Now these blue particles represent the information that was deposited by the mother. And how is that information received by the embryo? Well, if you can notice, there are these green protrusions coming out of the embryo surface, right? And each one of those green protrusions has a little bit of a blue spark on it. So each one of these green protrusions is a special protein which sits at the surface of the membrane. And that protein specifically acts as a receptor for the ligand that is out here. And so that ligand binds that receptor. And that receptor receives the information and passes the information through into the embryo. And so what I'm showing you here is something very interesting. So we are in an area where there's a lot of this outside surface signal, which is called a ligand. There's lots of ligand. And where we have lots of ligand, all of the receptors are bound to it. And you can see here this is an individual, but the ones that are bound are actually pairs. So what happens is, as the receptor binds to the ligand, the 2 individuals that would be floating around in the membrane come together and form what are called dimers. So that's what happens when you have a lot of signal around. Let's take us back out to the outside view and zoom out a little bit, because what I want to show you here is that you don't have the same amount of that signal all over the embryo. You have a lot of it at the tip here. If I move around and come this way, we have a little bit less here. And if I come out here, oops, if I come out here, we have less still. And then if I come out to the here, we have 0, none at all. And what's interesting is that something is corresponding inside of the embryo to that outside signal. Where we have a lot of outside signal, we have a lot of this red, sparkly action happening, right? So what does that mean? What is going on there? All right, well, let me show you. Let's go back to our high concentration of outside ligand to a lot of dimerized receptor. What happens on the inside of the cell when those receptors dimerize? Well, let's come into the inside, and what we can see here is that we have some complexes forming on the membrane. So what can I show you? This is what I'm choosing to represent as a protein, okay? And I'm going to call this protein, let's make it a little bit bigger so you can see it. Here we go. Here we go. I'm going to call this protein RAF, okay? Just know that that's what that protein is called. What happens is when the receptors bind to the outside ligand, there's a cascade of activation that takes place. This receptor is activated. It recruits proteins from the cytoplasm, such as these. They come in, they complex to the receptor, and they bring in the protein RAF, okay? That's RAF. It's brought to the membrane where the receptor is activated. And what happens when that receptor is activated and all of those proteins are brought together is that the information is passed along from the receptors through these proteins now, which talk to each other. And then finally, it comes to another protein which looks like this. And this protein is called MEC. And MEC is what I have chosen to represent as all of these sparkling red proteins that are accumulating inside the cytoplasm. Okay, so MEC comes to RAF, RAF activates it, and where you have a lot of, where you have a lot of active receptor, now you have a lot of active MEC, and you can see all of that now around me. Okay. So what happens then when you have a lot of active MEC? Well, ERK. MEC phosphorylates another protein called ERK. And ERK finds its way into the nucleus, where it can interact with other proteins. And so I'm choosing to represent that here, okay? And where we have a lot of active receptor, we have a lot of this protein now that's now in the nucleus, okay? And it's able to interact with another protein, which is called Capicua. And Capicua likes to sit on DNA, and it likes to occupy DNA and keep other proteins from binding to these places. Now, interestingly, ERK can come in to the nucleus, and when there's a lot more ERK in the nucleus than there is Capicua, ERK will bind to Capicua, it'll modify it, and then it will, kick it off of the nucleus, and you can see that happening here. And then once it's modified, ERK will let it go, and it'll get kicked out of the embryo, kicked out of the nucleus itself, and you can see it kind of accumulating in this space, right? And once that happens, once the Capicua is lost from the DNA, like so, now this DNA is accessible to another protein which can come in and can activate the genes necessary to make this cell have the right identity to make those head skeletal parts. Now, it's very interesting that we have this specific cell in this specific place of the embryo. Let me zoom back out. Okay, and it'll remind us that we are in an area of the embryo which has a lot of this external ligand, which activates a lot of receptor, activates a lot of the pathway, activates ERK, and a lot of that ERK can come into the nucleus and kick Kapikua out of that position of the DNA. What happens if I move over to a position now where we have even a little bit less extracellular ligand? Okay, let me just zoom down into the membrane of the embryo here. Okay, and we can see here, out here, there's a little bit less of the extracellular ligand and less of the receptor is bound to that ligand, and we have less dimers formed. If we move down now in through the membrane and into the embryo, we can see that because of that, there are less complexes formed. And then when we come into the nucleus itself, we can see that there's less ERK here now in the nucleus interacting with Kapikua. And so you can have more Kapikua bound to the DNA, keeping more of these sites of DNA closed off for activation. So now, as opposed to the cell that was over there, which in a space with a lot more ligand and a lot more activation and had the DNA open, here we have the DNA a little bit more closed. And so only one site, let's say, is accessible to an activator. And this cell might actually have a different fate than the cell that was over there. So what happens if I come a little bit farther out now and I take us to a port of the embryo where there is no external ligand? And we can see here, no external ligand floating around the paravital in space, nothing bound to the receptors. And so because there's nothing bound, they're all monomeric. And so they've not dimerized, no activation has occurred, that chain of events has not been passed along. And so there's no activated molecules. And then when we come into the nucleus, what do we find? We find that the entire nucleus is full of this other protein, Kapikua. It's completely covered the DNA. And now this cell cannot activate any of the genes that those cells on that side of the embryo were activating. And so what is happening here, if I zoom out again, is we have seen a gradient of information, which provides through a gradient of activation a different pattern of cell identities by affecting the availability of that strand of DNA to activating proteins. Here, that DNA is completely open, and so all of the genes necessary for making the head skeleton are going to be activated in that cell. As we go away from the source of the information, less and less of that DNA is going to be accessible to activators. And so different gene sets in each one of these cells are going to be turned on. And so each one of these cells is going to have a different identity. And then here we have a cell that's going to have a completely different identity, which is going to be completely different and apart from the head. Here we have different parts of the head. Here we have one that's not even going to be a part of the head. So this is a demonstration of a phenomenon called patterning. And patterning is very important because it tells different cells in the embryo to have different identities. And that's what tells the head to develop, or the head should develop. And that's what tells the tail to be where the tail should be. And parts that shouldn't be making head structures should make trunk structures. And so I hope this has served as a demonstration of two different scenarios that can be demonstrated through virtual reality, mixed reality. Obviously, my artistic skills are at a limit here. Different tools will hopefully be developed to allow for different assets to be brought into something like this. But this is just a kind of a quick proof of concept of what I would like to demonstrate here. And I hope you found it informative and at least entertaining. Thank you.